integrated lab medicine. So today we are going to talk about one important topic, that is amyloidosis. So what is amyloidosis? Friends, amyloidosis occurs when abnormal proteins called amyloids build up in organs and they impair their function. So as you can see in this pic, it affects multiple organs in our body like heart, kidney, liver and nerves. So let's understand the topic of amyloidosis. Now before that we take a look into the history of amyloidosis. So in 1639, the first time the lardaceous liver and white stone containing spleen was discovered. Later on in 1859, Friedrich and Kekule discovered protein called amyloid deposits for the first time. Then in 1927, bifringence enhanced by Congo red amyloid structure is identified. Now, then in 1978, there was first identification of the transthyretin as the precursor protein of familial amyloid polyneuropathy. Then in 2001, the ex vivo amyloid fibrils studied by cryo-electron microscopy. Then in 2013, the A beta nanotubes were discovered. And later on in 2017, the tau fibril structure was identified. Now let's move on to the next slide. Now let's talk about history. So the term amyloid is derived from the term amylum, which is a Latin word. It means cellulose or starch. Let me change the color and the pen. And the term amyloid was coined by Matthias Kleiden, who was a German botanist in 1838. The term amyloid was first used by Rudolf Virchow, as you can see in the pic, in 1854 because of the reaction with iodine. Later on in 1859, Carl Friedrich and August Kekule reported that the waxy spleen contained no material that corresponded chemically to the amylon or cellulose, as you saw in the previous slide also. Now, how is the amyloid formed? First of all, there is a term known as fibril aggregate. So, fibril aggregate is a tangled mass of amyloid fibrils of random orientation. Now, these are occasionally seen as parallel bundles and which are predominantly extracellular. Now, the next is amyloid fibril. Amyloid fibril is a double helix consisting of two pleated sheets micelles in the form of twin filaments separated by clear interspace, as you can see in this pic. Okay guys, now the amyloid filament. The amyloid filament is lozenge shaped Congo red or toluidine blue dye molecules, as you can see in this pic. These are Congo red dye molecules. They retained in grooved faces of beta pleated sheet protein by hydrogen bonds. Now amyloid, when you use X-ray diffraction method, the protein in the fibrils is arranged in an anti-parallel beta pleated sheet method. Now the main topic of the today's session is amyloidosis. So what is amyloidosis? Amyloidosis is a condition associated with a number of inherited and inflammatory disorders in which extracellular, not intracellular, it is extracellular deposits of fibrillar proteins that are responsible for tissue damage and functional compromise in the different organs of the body. Now, how is amyloid formed? The bulk of the amyloid deposits is formed of protein, or you can say it is composed of protein, the fibrillar proteins. And it also contains up to 15% of non-fibrillary glycoprotein, which is known as amyloid P component. Amyloid P component, that is AP, known as serum amyloid P component, SAP. It is very important term. Please remember this. Now, this SAP binds to the glycosaminoglycans, fibronectin, and other cellular components. And this binding to amyloid fibrils is used in patients with amyloidosis or for radio-labeled SAP scintigraphy, which is a diagnostic technique that is used for quantitative monitoring of amyloid deposits. Now let's move on to the ultrastructure of amyloids, which is seen in the electron microscopy. So here is the pic showing electron micrograph from the kidney, which is a case of amyloidosis with renal involvement, showing amyloid. This is the amyloid, guys. And epithelial cytoplasm, EpiC. This is the epithelial cytoplasm. And there is a basement membrane, as you can see, and an endothelial cell. This is an endothelial cell. So what is A? A is amyloid. EpiC is the epithelial cytoplasm. BM is the basement membrane, and endo is the endothelial cell. Now let's study this in some more depth. So these amyloid deposits appear as masses of extracellular non-branched filaments, usually in the random orientation. That is, these are arranged in zigzag pattern, not in parallel or some oriented manner. So each fibril, amyloid fibril, the total diameter of this is 8 to 10 nanometer with variable length of up to several microns. And you can see in the pic, it is a higher magnification of the previous image only. Now the next technique is X-ray crystallography and infrared spectroscopy. So in this diagram, it is a schematic diagram of an amyloid fiber showing 4 fibrils, 1, 2, 3 and 4. There can be as many as 6 also. These are wound around one another with regularly spaced binding of the Congo red dye. This is the Congo red dye which is present in the fibrils of the amyloid. Now this image shows the Congo red staining, and on the polarized microscopy the Congo red staining shows apple green bifringence, which is very important. This image is also very important. and it is a diagnostic feature of the amyloid. Okay, and in this pic you can see the electron micrograph of the amyloid fibrils. So that's all for today, guys. I hope you guys understood the topic of amyloidosis, and we will continue this topic in our next video as well. And don't forget to like, share, and subscribe my channel, and please press the bell icon also. Thank you, guys, and let's meet in the next video.